Okay, well, with that out of the way, I think I'll start sharing my screen. Can everybody see my screen? Okay. All right, thank you. So today's lecture will be on the digestive and pulmonary or respiratory systems. These may not be exactly connected, these two systems, when you think about it, and they're really not, but the reason they're grouped together is because I would say out of all of the systems that you have to learn for the MCAT in terms of biology, these are two of the most low yield. So usually respiratory and digestive systems aren't as heavily tested as, say, the renal system or cardiac system, muscular system, those kinds of systems, obviously nervous system. So that's why I grouped them into this lecture. So I still say it is very important for you guys to know these systems, and they're very important, especially if you do want to go into medicine, which I'm assuming most of you do if you're taking the MCAT. But they're the ones that are the least tested out of all of the biological systems in the body. Okay, this is just the consent to audio and video recording because this will be posted onto YouTube. A little quick personal introduction if this is your first lecture. My name is Mark Abdurrahmanov. I'm a graduate of Stony Brook University. I took the MCAT this January. This is my score with the breakdown. If you ever feel like contacting me with any questions or anything like that, you can always contact me personally at my email, which is located here. Quick description of this course. So it's a 15-week full course. I believe we're on week 9 or 10. I believe we're on week 10, actually. We have 3 lectures per week, Thursday, Friday, and Saturday, and office hours on Sundays. All events start at 8:30 p.m. Eastern Standard Time, just like this one. We also have a GroupMe. GroupMe has all of the lecture recording links and PowerPoints posted, as well as any updates or changes. It's also an easy way to get a hold of me or any of the other tutors. And there's also about 400 other students in there. So if you have any questions that you want to ask about the MCAT or about the process in general, or anything at all, please feel free to join the GroupMe and ask away. Again, me and all the tutors are there, as well as a bunch of students. So if there's a high likelihood that at least somebody will have the answer for you. Okay, so learning objectives for today, we're going to be learning about the digestive pathway. We're going to be learning about the important qualities of the digestive organs. We're going to be also looking at how does gas exchange occur in the pulmonary system, and what are the functions of the pulmonary organs other than gas exchange. Okay, so moving on to the content. First, we want to talk about the digestive pathway. So this is just a quick overview, and I'll go in depth into each of these steps. So each part of the digestive system has a role in digestion and absorption of nutrients. While not exclusively part of the digestive system, other organs actually are also part of the digestive system, including liver, pancreas, and gallbladder, because they also have roles in digestion. And the main digestive system would be the esophagus, the stomach, as I mentioned, the gallbladder, the small intestine, the large intestine, and then obviously the anus. So this is just a general, I would say, kind of pathway that it goes down, but we'll cover each step in more detail. So firstly, we have the mouth and the esophagus. So this is the first part of the pathway. The mouth is obviously responsible for mechanical breakdown of food with your teeth and just chewing, along with chemical digestion with saliva. Saliva contains the enzymes lipase and amylase, which break down complex fats and starches into more simpler lipids and sugars, respectively. Again, lipase is for fats into more simple lipids, and amylase is for starches into more simple sugars like glucose. And what we call this is, so the chewing part we call mechanical breakdown. This breakdown with enzymes like lipase and amylase, we call that chemical digestion or chemical breakdown, because we're essentially changing like the chemical structure of these molecules that we're breaking down. We're changing the chemical structure of the lipase, of the fats and starches using lipase and amylase into different chemicals that are easier for us, for our bodies to absorb and use. So digestion begins directly in the mouth. After the mouth, we go down into the esophagus. The esophagus is crucial because that is where chemical breakdown continues using the saliva. And it's also a way of mechanically transporting the food into the stomach and the rest of the GI tract. The food during the esophagus is packed into a bola, which you don't really have to know this, but it just looks like kind of like a log like this. So the food gets kind of compacted into like this type of shape, just like a log, I guess. Right, and that way it can travel very easily down the esophagus, which if you can see here is a very, I guess, more narrow pipe. So it needs to be more compact if it wants to travel down that way. So the first destination after the esophagus, which you can see ends right here, would be the stomach. And this is usually what we think of when we think of digestive system and just food in general. We just think it goes straight into our stomach. So the stomach is the first site of the first major site of digestion. I mentioned that digestion begins in the mouth and the esophagus, but the stomach is really the first main site. This is where some of the digestive enzymes reside, including pepsins, which are involved in the breakdown of proteins into small peptides to be recycled into the body. Right? We can make our own polypeptides and amino acids in our own body, but there are some amino acids, and it obviously is also more efficient to reuse them and get them from somewhere else. And the way we get them is from food. When you eat meat or any kind of food, really, it has proteins, and we want to use those proteins, but proteins tend to be very long, very complex, very big, and very difficult for us to use in their current form. However, we can break them down into smaller pieces and have small, short polypeptides and amino acids, and those we can utilize directly and recycle in our body. The way we do that is through these enzymes called pepsins, which, just like the amylase and the lipase, are specifically made for breaking down proteins. This is just one type of protein that's enzyme that's found in the stomach. There's also lipase, amylases, and other types of digestive enzymes. Most of these enzymes are called zymogens, and they're made in the pancreas away from the stomach. So these zymogens are made in an inactive state that cannot digest anything. The inactive state of pepsin, for example, is called pepsinogen. Once they are transported into the stomach from the pancreas, then they become active and can begin digestion. The reason for that is, obviously, the pepsin, which we're using as an example, it breaks down proteins. Well, if it was active as soon as it was made, it would just break down the proteins that made it, right? It would just break down any nearby proteins it sees, including the ones that are part of your own body. To avoid this, you make them in an inactive state in the pancreas, then you transport them in an inactive state where it's basically dormant, can't do anything. Then you transport it into the stomach, and then in the stomach where you actually want it to be, and to actually where you want it to start digesting stuff, that's when it'll be activated. So another function of the stomach is to produce acid, which is crucial for digestion. There are parietal cells in the stomach, and these are the specific cells that produce this acid. And they're found just on the lining of the stomach. So the stomach acid is extremely important for digestion. So the pH of the stomach is specifically kept very low and very acidic through the presence of HCl, or hydrochloric acid. The pH of the stomach is about 2 to 3. And this is because the digestive enzymes like the pepsin, which I mentioned, function best at a low pH. So this is actually one of the ways that we change from the inactive to active state. The rest of your body, the pH is always about 7.4-ish. So the enzyme pepsin at 7.4 won't really function because that's not its ideal environment. And because of that, it'll remain inactive. However, once it reaches the stomach where you actually want it to be and you want to activate it, the way you would activate it is just it senses the acidity in the environment and it becomes active and it functions best in that pH. So at higher pHs, they're essentially non-functional, like in the pancreas where they're made. This mechanism, like I mentioned, prevents pepsin from digesting the proteins in your own body. Oh, this actually, not sure why this is here. Sorry, this is a typo. So the stomach has an internal lining of mucus and bicarbonate, which protects the cells of the stomach from being damaged by the acid. If you see this bicarbonate, this is actually a base. So the acidic version of bicarbonate would be carbonic acid, H2CO3, right? This bicarbonate is this carbonic acid is the acidic form. This bicarbonate is the base form. Bases, as you know, neutralize acids. So the lining of the stomach has this mucus and this bicarbonate, which protect the cells of the stomach from being damaged by the acid. Obviously, the stomach walls are made of cells. Cells are not naturally resistant to acid. So the way you prevent them from just being burned and like kind of destroyed by the acid is you have to have some kind of protection. And this comes in the form of mucus and bicarbonate. The mucus acts as a physical barrier. If you know what mucus is, it physically stops the acid from reaching the cells, right? Because it's a viscous liquid. So it prevents it from actually making contact and dissolving it. And the mucus also is mixed in with bicarbonate, which helps act as a chemical barrier, because as soon as the hydrochloric acid makes contact with the mucus bicarbonate mixture, it'll become neutralized and essentially non-acidic anymore and not damaging, right? So you have a base and the mucus, which acts both mechanically and chemically to prevent damage to your lining of your stomach. If you guys know about acid reflux, so where you can have, it's also called heartburn sometimes. So it's not a buffer in the sense of chemistry buffer. It's a buffer in like the real world sense, where it's like it buffers something. So or it acts like a barrier. In that case, the buffer. In the chemical sense, it's not a buffer, because buffers are really there to resist changes in pH, whereas this is meant to really neutralize instead. So it's a little bit different. Yes, it's not exactly a buffer. So somebody asked in the chat, not in the chemical sense at least. But like I mentioned, there's acid reflux, or sometimes called heartburn, because it feels like it's in your chest. So this all is protected, right, by these cells that I mentioned in this mucus. However, and you imagine this is all filled up with the acid, sometimes with people, they have the acid come up, and see, called the esophageal reflux, can come up into this esophagus. The esophagus is obviously not protected by this mucus and this bicarbonate. And so the cells over here in the esophagus begin to become dissolved by the acid. And that's why you feel like the chest pain or just the pain if you have acid reflux or heartburn. Okay. And this is, like I mentioned, it's a typo, but what I did want to mention is that the acid also acts to kind of destabilize any protein. So proteins obviously have 3D structure. They sometimes have function when usually when you cook something, like if you are making meat, when you cook it, it'll denature the proteins, obviously, which will render them non-functional and unwind them. But if you're eating something that's raw that has proteins, the acid also acts to denature them through chemical means, and that's what was supposed to be written here. And I'll make sure when the PowerPoint goes up, I just want to mention it. It's not extremely important, but just for you guys to know. So the next step after the stomach, and you guys can see here, So it came from the stomach. Now it's this really, really long tube-like thing called the small intestine. So the small intestine is the main site of digestion. The stomach was kind of the beginning, and it was the first main part of digestion, but the small intestine is where the vast majority of digestion occurs. It has a huge surface area due to its just many layers of folding. So you can see, firstly, it's really, really long, and it's folded on itself like this way, right? Which gives it a lot of area, and it's a very long like tract. But if you look at this close up right here on the actual like tubing, it has these small little finger-like things called villi, and this is a even more zoom in of that. So these little villi. What this means is that the actual tubing is not just smooth, right? It's not like a smooth tubing. The actual tubing itself is made of these little like ridges and like finger-like appendages, and that adds just a lot of surface area to it. So I think I remember in undergraduate when I learned this for the first time, my professor mentioned that if you took a small intestine and you like opened it out completely, removed any folding, removed all of like the villi, completely stretched out, it would be like the size inside your body would be like the size of a football field or something like that. So that just goes to show how much actual area is inside of your small intestine. And if you guys remember from chemistry, higher surface area means more space to kind of react and go and kind of reabsorb through diffusion, for diffusion to occur, for the reabsorption of nutrients. It also means a lot of area for these enzymes to kind of work with. So the huge surface area of the small intestine allows maximum digestion with enzymes, and for very good reabsorption of the broken down nutrients into the surrounding bloodstream, to surrounding capillaries. So that was just the main thing about the small intestine. That's like the main point of digestion. That's where the vast majority of digestion will occur. So the breakdown with chemicals, like the enzymes, and the absorption into your actual body. The next area that we want to talk about is the large intestine, also called the colon. This one's not really as heavily tested. It's not as important. The role of the large intestine is really to complete reabsorption of any water and nutrients left. So after the small intestine, most of it will have been absorbed into the body, but not all of it. The colon is just there to finish reabsorption. And once you're done with the colon, that's essentially the last step. And because of that, ideally, you want to have all of the water and all of the nutrients that were in the food be absorbed, and the only thing you're essentially left with is waste in the form of feces, which will be excreted through the anus. So the colon is kind of like the final, I guess, checkpoint where you just want to make sure that everything you can get that's useful from the food can be taken from the food. And then after this, it's sent to the anus for excretion. We're not really going to talk too much about that because it's not very important. Something that I did mention a little bit is the gallbladder and the liver as being part of the digestive system, and I wanted to cover how that is. So there's this substance called bile. So bile is produced in the liver and it's stored in the gallbladder. This is one of the main actual functions of the gallbladder. It's secreted from the gallbladder into the stomach during digestion of fats, right? So like I mentioned, lipases are very good for chemically breaking down fats. However, some fats are too big to even be broken down with lipases. So what you need is this bile to emulsify the fats, or kind of mechanically or physically break them down into smaller pieces. And then the lipases can chemically break them down into simpler fats, right? So this doesn't actually make the fats more simple, it just makes them smaller into smaller chunks. Whereas lipases actually make them simpler fats chemically, right? like I mentioned, emulsifies fats by breaking large chunks into smaller fatty acids, which are easier to digest through lipases. Okay. Now that I've been talking for a while, before we move on to our first question, does anybody have any questions for me about anything that I've covered so far, anything about the digestive system? Before we move on to our first poll question of the day. Okay, if not, I'll move on. So the products of the liver participate in blank digestion, and the products of the pancreas participate in blank digestion. And I just want you guys to fill in the words like in order, right? Okay, polls should be up if you guys want to answer A, B, C, or D, or if you really don't know, pick I have no idea. Okay, take another like 30 seconds or so. All right, so I'm gonna end the polling. So the majority of people picked answer choice C, with a couple people picking A. So correct answer is actually B. So, and I kind of expected this because this is a little bit of, I guess, a tricky question. So think about what are the products of the liver and what are the products of the pancreas. So that's the first step. Does anybody want to put in the chat what the products of the liver are? I just mentioned it recently, but if you guys want to, anybody wants to put in the chat what the products of the liver that participate in digestion are. Right, bile. Even though it's stored in the gallbladder, technically it's made in the liver, so it's a product of the liver. And what are the products of the pancreas? So this has a couple of different names, I guess. So it's not fatty acids. Fatty acids are what the fats are broken down into. Yeah, lipase, or more generally it's zymogens, but lipase is one of them, yeah. So lipase is one of the enzymes that gets produced in the pancreas. So it does, you may think that bile breaks down fats, emulsifies fats and breaks them down into fatty acids, smaller fatty acids, and lipase breaks them down into simpler fats that we can use, right? Well, like I mentioned, bile, when it emulsifies fats, it doesn't actually chemically change the fats. It just physically breaks them apart. So like if you had, say, a sandwich, and then you took a knife and you cut it in half, you made a smaller sandwich, but it's still the exact same thing. It's still the same bread, the same meat, same everything. Everything's still the same. You just made it smaller. You didn't chemically change it. You physically just made it smaller. That's sort of what bile does, right? So it's not actually chemically changing it, it's physically changing it. Whereas lipase, if you took that sandwich and you threw it in some acid, and then that sandwich is no longer a sandwich, something completely different. It's chemically completely different now, right? So that's kind of why it's mechanical and chemical. So this would be, the bile would be mechanical, the pancreas, the lipase would be chemical. Does that kind of make sense to everybody? Okay, if there are no questions, and if you have any questions, please feel free to ask them in the chat. But if not,
Yeah, quick question with the pancreatic cancer. They are not allowed fatty foods because there's no. Why are they not allowed fatty foods if you have pancreatic cancer?
Well, if you have pancreatic cancer, that means so obviously this is a little bit more in depth and more challenging. But what I would assume is because if the pancreas is not functioning, you wouldn't be producing a proper amount of the enzymes, the lipase, the pepsin, the whatever. You wouldn't be producing enough of that in order to actually break down the fat, and so the fat kind of it's more likely to get like clogged up and stuff, right? Because it still remains in its in complex form.
Okay, so you're not producing the enzymes to break down the fats.
That could be one. That would be what I would assume. I haven't done research on this,
Yeah,
so
that
I
makes
wouldn't.
sense.
But yeah, but if you have, this kind of relates to our next question. So actually, if you want to answer this first, but if anybody has anything else, please.
Yeah, that makes sense. That makes sense. Yeah.
Okay, so please take a little bit of time to answer this question. The poll should be up. Okay, take another 15, 20 seconds to answer this. Okay, and then the polling. So the results are fairly mixed, with D having a slight lead. However, the correct answer is C, and I'll go over it. Right. So if patient has a condition which causes an increased amount of fat in the feces, which organ is least likely to be affected? So whenever you see a question like this, the first thing you want to think of is, what is this question actually asking? So if the patient has a condition which causes an increased amount of fat in the feces, that means the fat is not being absorbed into the body, right? And there's a number of ways that this could happen. And actually, 3 of these organs will present 3 different ways that this could happen. And one of these will really not be involved. So what you're really looking for is which organ is not involved in digesting and absorbing fats. Well, we just mentioned the liver makes the bile. Bile is needed to emulsify fats to make them easier to break down with lipase. So liver is very important for breaking down fats. So it can't be A. It can't be D, because D, actually, the pancreas is what makes the enzymes that break down the fat, the lipases. So if you don't have the lipase, you can't digest the fat chemically. So it's between B and C. So what are the functions of the small intestine and stomach in relation to fat? Both of them are sites of digestion. And the small intestine specifically is the site of majorly the site of digestion and reabsorption, right? The stomach doesn't have any reabsorption. That's all in the small intestine and then the colon, right? Because you really do need the small intestine, because that's where most of the fat will be digested. and that's where most of the fat will be reabsorbed into the body. So if there's an issue with your small intestine, that means that you're either not digesting the fat or you're not reabsorbing it into the body after it's been digested. However, the stomach is involved in some digestion of fat, but very little, not as much as the small intestine. The stomach is mostly there to produce acid and just begin the digestion process, denature any proteins and anything like that, right? So out of these 4 options, the stomach is the one that's least involved in digestion and reabsorption of fats into the body. So that's why it would be the correct answer. Any questions before I move on? Please feel free to ask in the chat. Otherwise, we'll move into respiration, which is the next topic for today. So I assume all of you know the main organ in respiration are the lungs, right? Lungs are a very complex organ, and the MCAT doesn't really test too much on it because parts of it are really complex, and it's just not as something that's not very popular with the MCAT. As you can see here, there's many different things that are labeled here. Really, you don't have to know. The majority ones that you have to know is the trachea, which is kind of like which brings in air from the mouth into the lungs, right? Branches off here and here into the, obviously have 2 lungs. Most people have 2 lungs, and they should look symmetrically. They have these branches. The branches are called bronchi, right? These little branching, like tree-like things. And then at the end of these branches, right here and here and here, everywhere, there are these small, bunch of these small sacs called the alveoli. And this is like the most important part of the lungs, the alveoli. Okay. And then under the lungs, you have this big pink muscle called the diaphragm, this dome-shaped muscle. And this is extremely important. That's how we breathe, is through that muscle, the diaphragm. So the diaphragm is a muscle that is the driving force behind respiration. When the diaphragm contracts, we inhale. When it relaxes, we exhale. So if you want to breathe in, you're contracting the diaphragm. Then when you breathe out, you relax the diaphragm. When the diaphragm contracts, it pushes the rib cage out and it expands the lungs, like physically, like a balloon, it expands it. When it relaxes, the volume returns to normal. This next section will be a little bit of a throwback to the fluids section where we mentioned gas and pressure. But when you expand the lung volume and reduce the resistance, you actually decrease the pressure in the lungs, right? The atmospheric pressure in the lungs gets decreased as a result whenever you contract the diaphragm. So, and as we mentioned in the fluids lecture, gas always flows from high pressure to low pressure environments. This principle is the reason we inhale and exhale air. Inhaling is just the gas moving from the high pressure outside to the low pressure inside. Exhaling is just air moving from the high pressure inside to the low pressure outside. When the diaphragm contracts and lung volume increases, the pressure in the lungs decreases lower than that of the outside atmosphere. This creates a pressure gradient, which creates a sort of vacuum that causes the air in the environment to rush into our lungs. If you want to think of it, when you actually do like a sucking thing, like when you suck in air, your body's not actually doing anything to bring the air in. All you're doing is just making your lungs bigger. And essentially the laws of physics do the rest. You're not actually doing anything to move the air from the outside to the inside other than expand your lungs. And then naturally the air will just flow from the outside to the inside. It'll kind of like rush in and you can't really do anything about it. This is the same principle, like if you're flying in a plane or a spaceship or something, the pressure inside the plane or the spaceship is much higher than the pressure in very high in the sky or in space. So if you open the door in a plane while you're flying, or if you open the door in a spaceship while you're in space, all of the air and everything inside the carrier will just immediately get sucked out into the atmosphere. This is the exact same principle. You create a pressure gradient, and then the air just rushes in and kind of goes down its gradient from high pressure to low pressure. During exhalation, the pressure in the lungs returns to normal, which is higher than the pressure outside, and the air flows down its pressure gradient out of your lungs, right? However, with exhalation, it's a little bit different because your body can physically assist in exhaling during strenuous exercise, or when you are just forcefully breathing and you're trying to breathe in a lot and really fast. Your body actually helps. There's some muscles in your abdomen that will kind of like push the air out more, basically. That's only when you're exhaling, not when you're inhaling. But it's normally when you're just talking, breathing normally, it's also based solely on pressure gradients. Only under like high, like forceful breathing is it really like your abdomen also helps. So I mentioned these things called the alveoli. So the lungs have many different branches called the bronchi, which end in small sacs called alveoli, which hold all of the oxygen until it is passed into your blood. These alveoli have changing air pressures. So if you looked at a graph of pressure over time in the alveoli, it would look something like this, right? Oh, sorry. Look something like this, and then it would go like this, then back up, then like this, like that, right? It would be always changing. When you're trying to inhale, the alveoli have very low pressure to allow the air to come in, down its pressure gradient. When you want to exhale, the alveoli have a lot of pressure, and you want to get it out into the environment, right? Go down the pressure gradient. So inhalation, low pressure inside the lungs, exhaling, high pressure in the lungs, basically. And that goes with the alveoli as well. The air is then rapidly diffused between these alveoli and the blood in nearby capillaries which pass by them. So, like I mentioned, the alveoli are very, very small, like circular sacs, and there's a bunch of them, like a lot of them. And the capillaries are also very, very thin, and there's a lot of capillaries that kind of reaching around. Because when you have something that's very small and a lot of it, that actually creates a lot more surface area than if you just had one big tube, right? Having many small tubes is much more surface area than having one big tube. The large surface area of contact between the alveoli and the capillaries make the process of exchanging oxygen and carbon dioxide very, very fast, and you can do it very efficiently. That's why you can breathe very quickly, and like the air can transport very fast. Right, and here's a diagram here on the right. As you can see, these sacs are the alveoli, and then these are like capillaries that kind of surround it, right? Okay, so moving on. Another important thing for oxygen transport in the blood. Now that the oxygen is in your blood, we want to look at this molecule called hemoglobin. Hemoglobin is a complex molecule which you don't need to know the structure of, I believe, but it's just a very big molecule with a lot of different parts to it. It's a complex between like protein, like actually multiple proteins, as well as involves iron as well as like a cofactor, but it's just, you don't really have to know it. Just know hemoglobin is mainly used for transporting oxygen. It's a complex molecule found only in, or mainly in red blood cells, and it acts as a carrier for oxygen and carbon dioxide. Once the blood reaches the lungs, the oxygen diffuses through the membrane and into the vessels or in the capillaries, like I mentioned. And when it does that, the oxygen binds to the hemoglobin in the blood, as oxygen has a very strong affinity for hemoglobin. Once the blood reaches its target tissue, the blood bound to the hemoglobin will unbind and go into the cell. The process of how it does this is a little bit complicated, and I don't believe they really test on it in the MCAT. If you took a biochemistry class, you probably have talked about this whole process, but for the MCAT, it's not really important because it's a little bit in depth, right? But yeah, the main idea is that hemoglobin is kind of like the carrier. If you want to think of it, it's like a taxi almost for your oxygen. It picks up the oxygen at the lungs, transports them to wherever it wants to go, the arms, legs, whatever, liver, and then it transports it there, drops off its passenger, which is the oxygen, and then goes back to the lungs through the system, like through the heart, goes back to its and picks up a new passenger. Right? Do we need to know how many Fe it binds, like a tetramer or not? So not really, no. Know that hemoglobin, maybe you may need to know that hemoglobin is kind of a tetramer, but the main thing you actually have to know about hemoglobin structure is that it has a quaternary structure. So if you remember from proteins, they have a primary structure, secondary structure, tertiary structure, and then some have a quaternary structure. Whenever anybody in any class pretty much gives an example of a quaternary structure, they pretty much always say hemoglobin, because that's the main example in your body that everybody knows and is easy to talk about. It just means that there's multiple subunits in hemoglobin that act as a tetramer. I believe it binds one iron, but you don't really have to know that, and the structure is way too complex, right? So don't worry about it, but just know it uses iron. Okay. So there's this binding curve, which you may have to know for hemoglobin, and there's a lot I want to cover here. So firstly, look at the y-axis. Whenever you see a graph, just on the MCAT or anywhere, you'll see a lot of graphs. And whenever you see graphs, I always suggest first thing, look at what is the y-axis, what is the x-axis. That'll tell you basically what we're measuring and what we're even looking at here. The y-axis is the hemoglobin saturation. What that means is what percentage, and you can see a percentage sign, what percentage of hemoglobin is currently bound to oxygen, right? I said that it has a high affinity, and so it'll bind to oxygen, but it's not just like instant, right? Nothing in chemistry is really like that. So it's like a gradual change, and it doesn't always get fully saturated, but this shows you, the y-axis shows you the percentage of hemoglobin that is bound to oxygen at this current moment. On the x-axis, we have this really stands for the partial pressure of oxygen, but really what that could mean, and what you should interpret it as to make it simpler, is just the amount of oxygen that is present in like the environment or the air, right? Or in like the space, right? How much oxygen is actually there. So if you go to the right, you have more oxygen in the space with the hemoglobin. If you go to the left, you have very little oxygen in the space with the hemoglobin, right? And this is what this graph is measuring. So as you increase the amount of oxygen available, how much of the hemoglobin will be bound to oxygen? As you expect, you increase the amount of total oxygen available, more and more of the hemoglobin will be bound to oxygen, a higher percentage, right? And that makes sense. First thing you guys should look at is this normal line, which is in red. This is what it normally looks like. This is normally what a binding curve looks like for hemoglobin. An important thing I want to mention, and if you guys remember this from the Michaelis-Menten curve, right? It's very similar to Km value. It's called a P50. A P50, what that means is at what pressure or what amount of oxygen would exactly 50% of the hemoglobin be saturated or bound to oxygen. So remember that the P50 is an amount of oxygen. It's this, right? It's the quantity, the units of it are this essentially, right? And I'm going to clear my drawings, but P50, it's actually just the units of this, right? It's how much oxygen is there. That's an important thing I really want to like stress. That's why I'm saying it a couple of times. It's how much oxygen is there. It's an amount. What what's special about this specific amount is this is the amount that will cause exactly half of the hemoglobin to be saturated with oxygen and half to not be to not have oxygen, essentially, right? That's what we measure as, and just like we use Km in Michaelis-Menten kinetics to measure the affinity, we use the P50 value to measure the affinity of oxygen for hemoglobin because it changes depending on the conditions, right? So if you have a low P50 value, that means you need very little oxygen in order to saturate a lot of hemoglobin, meaning high affinity. If you have a high P50 value, that means you need a lot of oxygen in order to saturate the same amount of hemoglobin, meaning you have a low affinity. Okay, low P50, high affinity, high P50, low affinity, low P50, high affinity, high P50, low affinity. Okay. Now, moving on from that, I now want to mention this green line and this blue line called the right shift, left shift. The reason it's called the right shift because if you took this red line and you moved it to the right, you would have the green line. Left shift, you took the red line, the normal, you moved it to the left, you have the blue line. That's why it's called the right shift, left shift. The right shift would have a higher P50 value, right? That's the main thing. So if the P50 is on this line, right? So this is 50% saturation. So if you went here and now you went down, so now you see the P50, which I said in units is amount of this, right? This is the units. Now it's higher, say it's 54, right? Just as an example. So now, in order to saturate the same amount of hemoglobin, you need twice as much oxygen available. That means it's half, the affinity is halved essentially. Left shift is the opposite. The P50 is over here. So the P50 is over here. Say it's 15. Now you only need half as much oxygen. Therefore, the affinity is twice as high. Right? Hopefully that makes sense. Right shifting. Just remember, right shifting is always shifting the graph physically to the right, which increases the P50 and lowers the affinity. Right shift is lowering affinity. Left shift is increasing affinity of hemoglobin to oxygen. Okay. Now, under what conditions will a right shift or a left shift occur? Right shift, mainly, at least for MCAT purposes, right shift will occur in high acidic environments. So low pH or high H+ means acidic. In the body, the main acid that you have in the body, and I'll talk about this actually in a second, the main acid that you have present throughout the body is actually CO2, is actually carbon dioxide. That's the main acid that you have in your body. So having a lot of carbon dioxide in your body will actually decrease the affinity of hemoglobin to oxygen, and it will make the whole body, it will make your pH more acidic, right? On the other hand, if you decrease the pH, or if you decrease the acidity, or increase the pH, make it more basic, you'll have a left shift. You'll increase the affinity, right? Other ways are temperature, obviously, but you don't really have to, it's not as important, but temperature, right? To heat something, less affinity. To cool it, a little more affinity. But this one, DPG, this may come up. I very much doubt it will. If you took a biochemistry class, you probably have talked about DPG, but I very much doubt you'll see it on the MCAT. It's a molecule that is present specifically in people who live at high altitudes and people who have adapted to live in high altitudes. They will have this molecule like DPG. They'll have they would have evolved to have more of this, and it's for specific reason because the air up there is thinner, but really, you don't have to go into that. Like, but if you are curious, look into it more on your own. But I would not, like, I'm not stressing it too much, right? And these are not important. Like, these are not really important. These are not really that important either. At least for the MCAT purposes. Like in real life, they're obviously very important, but for the MCAT purposes, they're not.
Mark, the DPG stand for the diphosphoglycerate. Is that
I believe so, but I actually don't remember what it stands for. But it's a molecule that's that what I remember is it's a molecule that's present in people who live at high altitudes where the air is thinner, and it's specifically used because if you didn't have it, it would be very, very difficult for you to get oxygen saturation in your cells, specifically at high altitudes. That's why people who are not adapted in such a way, like if you tried to climb Mount Everest, it would be very difficult for you compared to somebody who normally lives at high altitudes. Okay, so do want to mention that by increasing the affinity of oxygen to hemoglobin, yes, it becomes easier for the oxygen in your lungs to go into your blood, but it also decreases the oxygen unloading, which is actually getting the oxygen to your cells, right? Because if the oxygen is very, very high affinity for your hemoglobin in your blood, when it reaches the cells and it's time for it to get off, it won't get off because it'll just stay on. So it might be harder to unload it. So if you have too high of an affinity of hemoglobin to oxygen, then you may have problems getting oxygen to your cells. You'll have a lot of oxygen in your blood, but not a lot in your cells, right? And that's a common problem that some people have who have like blood disorders. Okay, and right, and then obviously if you have too much of a right shift, then it'll be difficult to get the oxygen from your lungs into your blood, which it will also eventually get in. Yeah. Yeah, left shift is a more basic environment. Yeah. Okay.
Mark, quickly, during the monoxide poisoning, what's going on what's going on with monoxide poisoning? Is it that a left shift where it's not unloading?
That would be an interesting topic for you to actually look up. So after you like learn this and kind of like figure this out, try looking it up and seeing if you can figure it out.
Okay. All right.
That would be an interesting thing to kind of like do a little bit of research on your own. And I highly encourage, like, when you have like these questions, oh, what about like pancreatic cancer? Oh, what about this? Well, I'm not always gonna be able to like give you like answers like this. What I want to actually do, like more than anything, is to teach you guys like the basics, and schools like the same way. We will like for you guys to learn the basics and learn the actual principles behind it. And then whenever you have this question, oh, what about this? Oh, what about this situation? Oh, what about that? You can actually look it up, and then you'll be able to understand it and kind of connect it, and that'll help you form more meaningful connections to this. Because like a right shift, left shift, this doesn't tell you that much. But if you remember, if you do some research and go into monoxide poisoning, that will help you kind of like learn it better, right? So I highly encourage you to, and everybody to, if you have any questions that come up as you're like thinking about like real life scenarios, please do some research and try and see if you can connect it, right?
Okay, thank you.
No problem. So last thing I want to touch on before we get onto the clicker questions is lungs for pH regulation. So along with the kidneys, which are also an important pH regulator in the body, the lungs are the other main pH regulator in the body. This is because one of the main sources of acidity in our body is CO2. So if you guys remember from acids and bases, CO2 is not inherently acidic, right? It's not inherently a Lewis acid or a Bronsted, whatever, Lowry acid. But in our bodies, CO2 reacts with water to produce this, H2CO3, right, which I actually mentioned earlier is carbonic acid, right? This is also an acid, right? And this is made through CO2. So if you have a lot of CO2, by proxy, you'll have a lot of carbonic acid in your body because you always have a lot of water in your body, which will react, right? This carbonic acid is acidic. Thus, having high levels of CO2 causes our pH to lower kind of indirectly, right? And just for reference, the conjugate base of bicarbonate I already mentioned is of carbonic acid is bicarbonate, HCO3 minus, which is also very important for you guys to remember just about what bicarbonate is. And it's a base, obviously the conjugate base. And then obviously, since the lungs are the main way to regulate the amount of CO2 in your body, it's a major pH balancer. If you're breathing a lot and heavily, you're going to be getting rid of a lot of CO2. Getting rid of a lot of acid means your pH will actually go up, and you'll have your blood will be more alkaline, or your body will be more alkaline compared to normal, right? If you're breathing very poorly and you're not taking a lot of breaths, what could actually happen is acidosis, which is when acid starts to build up in your body because you're not breathing enough, and not enough CO2 is being expelled, and you'll have kind of acidosis in your body, and you'll have your body pH will drop too much. Okay, and obviously both ways are bad. If you have too much alkaline or too much acidity, proteins stop working, enzymes stop working. Ideally, cells start to have problems, and it's not good. It's just not good either way, right? But yeah. Any quick questions before we get on to the poll questions for today? Okay, so please take a minute or so to answer this question. Okay, take another maybe 15 seconds to answer. Okay, and pulling. So actually 100 people, 100% of people said B, and I'm happy you guys got it, simple diffusion. I'll just go over it quickly. So osmosis, this is automatically wrong. Osmosis is only for water, right? And I want you guys to kind of like keep that in your memory, like at all times. If you're dealing with water, osmosis is the main method of transport of water. If you're not dealing with water, osmosis is automatically always wrong. So looking at the other three options, what is the mechanism of oxygen transfer from the lungs to the blood? Like I mentioned, it goes from the alveoli to the bloodstream through the membrane, to the capillaries through the membrane, right? Active transport means that you have to expend ATP, usually to go against the gradient. That's what active transport is usually used for, to go against the gradient. In this case, like I mentioned, you're going with a gradient with like oxygen. There's high oxygen in the alveoli, low oxygen in the capillaries, right? Because in the blood, it's not oxygenated blood, it's deoxygenated blood. So the blood doesn't have the oxygen yet. That's why I'll go there. So it's not active transport. Simple diffusion and facilitated diffusion. The main difference between these is simple diffusion is directly across a membrane. So if you have space here and space here, and then you just have a membrane, which is a wall, and then this can directly go here and this can directly go here with no problem. That's simple diffusion. If you had the same situation, you had a space here and space here, you had a wall. However, in facilitated diffusion, this stuff cannot go past this wall, and this stuff cannot go past this wall. You need special channels, or like membrane channel proteins or something like that, to actually facilitate the diffusion across the membrane, because it wouldn't normally be able to go through the membrane. In this case, oxygen's a fairly small molecule compared to things like sugar, proteins, and things like that. Oxygen's fairly small, so it has no problem diffusing just simply across the membrane. It does need to go through channels. Okay, so that's why when osmosis is not water, active transport is going with the gradient, not against. Facilitated diffusion doesn't use any membrane, doesn't use any channel proteins or anything like that, or carriers. So he's the right answer. And this question. So any questions about the previous problem? Okay, if not, I'll start the polling. Take another minute. Okay, take another minute or so to answer this. Okay, take another maybe 30 seconds to answer this. This one's a little bit more in depth, but I think you guys can do it. All right. I'm gonna stop the polling. Okay, so people chose choice A and C. So the correct answer actually is choice A. So which of the following occurs during expiration? So increase in lung volume will be eliminated first, because increasing the lung volume, it actually happens during inhalation or inspiration, right? When you breathe in, like I mentioned, ribs get pushed out, lungs expand. That's what causes the drop in pressure in the lungs. So it wouldn't be increase in lung volume. Actually the opposite would happen. They would deflate. Contraction of diaphragm again happens during inspiration rather than expiration. Contraction of the diaphragm causes lungs to expand, lets you breathe in. Right. However, A, increase of pressure in the lung relative to the environment. Again, increasing in lungs relative to the environment, that creates a gradient. Following air follows gradient and goes out. Okay. Contraction of external intercostal muscles. So this I didn't really cover too in depth, but these are just kind of like the other muscles in your chest, abdomen area that are also used for, mainly they're used for like forceful breathing, but they aren't really used for normal breathing, right? So that one would be the answer, and A is, out of these, A is the best answer. So any questions about anything we covered today at all?